Welcome Genetic Teacher Channel. Today, we are talking about biological lab supplies, which are tools made from glass or plastic, as mentioned inside slide. Safety goggles, protect eyes from burns. Light microscope, used to view small objects. Thermometer, used to measure kinetic energy of molecules in an object. Erlenmeyer Flask, used to mix chemicals. Volumetric Flask, used to make solution buffer. Florence or Boiling Flask, tolerate high temperature. Beaker, used to store buffer. Reagent Bottles, used to store chemicals and buffers. Beaker Tongs, used to pick up beakers and flasks. Stirring glass rod, used to stir liquid mixture. Graduate cylinder, used to measure quantities of liquid. Funnel, used to transfer liquid for filtration. Test tube, used to store liquid chemicals and microbial cultures. Test tube clamp, used to hold test tube. Test tube rack, used to store test tubes. Test tube brush, used to clean test tubes. Rubber stopper, used to plug test tubes and Erlenmeyer flasks. Centrifuge and sample tubes, used to load samples for biological reactions. Centrifuge and sample tubes rack, used to store centrifuge and sample tubes. And this slide show how manipulate with centrifuge and sample tubes during apply biological reactions, such as DNA extraction and others. Well plate, used to minimize chemical usage. Petri dish, used for microbial culture and chemical reactions. Wash bottle, used to wash glassware. Bunsen burner, used to heat chemical buffers. Wing top, used to spread Bunsen burner flame. Wire gauze, used to spread Bunsen burner flame. Ring stand, used to support rings and clamps. Ring, used to support beaker and funnels. Clay pip triangle, used to support beakers or flasks on ring stand. Burette, used to dispense quantities of liquids. Pipette, used to transfer amounts of liquids. Pipette pumps and bulbs, used to fill glass pipettes. Dispenser, used to deliver liquids. Micropipettes, used to dispense volumes of liquids. Dropper, used to transfer amounts of liquids. Mortar and Pestle, used to grind solid matter to powder. Evaporating Dish, used to boil off liquids, for leaving dissolving solids. Spatula and Scoopula, used to dispense solid and powder chemicals. Forceps or Tweezers, used to manipulate small objects. Scissors, used to cut tissues. Scalpel, used to cut tissues. Probe, used to remove underlying tissues. Dissection pins, used to secure specimen to dissection tray. Dissection tray, used to store specimen for dissection. Finally, this is biological lab structure. Thanks for attention. Please like and share our videos, and subscribe Genetic Teacher channel, and for any questions, please comment us.